Great to be here. Thanks so much for having us.
No, it's an absolute delight. Thank you for coming and talking about. So MouseView is a mouse tracking alternative to eye tracking experiments to get better accuracy when you're doing experiments online. Is that about right?
Yeah. It's a alternative way of scaling up eye tracking for online research that actually doesn't involve people's eyes, at least not directly.
Perfect. Well, I'm going to leave this session all over to you. I'm going to turn my video off. Off you go, Tom.
All right. Well, I'm going to hand things over to Alex, and he's going to talk about the validity of this eye tracking substitute and how you can use it on different online platforms, including Gorilla.
Okay, just give me a second to start sharing my screen. Okay. Cool. Okay, so thanks for joining, everyone. Welcome to the specialist seminar on our MouseView.js library. I'm Alex Irvine. I'm just coming to the end of my PhD at the University of Cambridge, and I currently work as an R&D intern at Cambridge Cognition, and I was also a part-time developer at Gorilla. Myself, Tom, and Edwin made this library, and most of the software development has been done by myself, which is why I'm going to give this talk today about the library. And much of what we'll discuss is also detailed in our preprint on site archive, which you can check out on our website. Okay, so just to give a brief outline, this talk will be in three main parts. So the first will be a bit behind where we thought this tool would be the answer to some of our research challenges. The second will be about how the data gathered using MouseView compares to a typical free viewing eye tracking task. And then thirdly, I'll talk about how you actually add this to an experiment using Gorilla's Zone as an example. Okay, so firstly, why did we decide to make this? As the pandemic closed multiple labs this year and last year, eye tracking became difficult to do in a typical way. And the obvious alternative was to use webcams to do this, which generally speaking is a great option. There are plenty of tools out there like WebGazer, which has been built into platforms such as Gorilla and JSPsych and others. However, we found solutions like this tend to amplify the issues that you experience with eye tracking in the lab. And these are things like losing the eyes, or issues with finding landmarks on the face, or contrast problems with the camera and eyewear. And further to the list, you get lots of attrition, which we have to combat with large samples. And then even after that, we end up with relatively low resolution data, which for certain research questions is interesting. But for us and our collaborators, this was quite difficult to work with. And so we thought, wouldn't it be great if we could improve upon these accuracy issues with a different tool? And would it be even better if we could avoid the awkward calibration stages and those errors too, as these are the source of much of the attrition that you can experience when running these types of experiments online. And the solution we came up with is mouse tracking with an occluding layer. And this is a pretty simple idea, and it has none of the fiddly technical issues that you have with webcam eye tracking. And this works in the following main steps. So first, we create an overlay atop any website, and this can either be solid or a partial cover. Second, we then detect the mouse position or touch position on the touch screen, and then record those coordinates. And at the same time, an aperture is cut in the overlay, which reveals the content underneath. We then iterate between points 2 and 3, which moves the aperture to the mouse location or finger location on the touch screen every time the screen refreshes. And this forces the user to move the mouse to any area that they're interested in. And over time, we can build up an attentional map, which is analogous to those that we make with eye trackers. And to drive this point home, here's an example of maps we produced using eye trackers on the top, and then the equivalent map just using MouseView.js. And even though this is just collapsed over time, you can see that they're pretty similar topologically. And this is what MouseView looks like in practice. So the end result is here. And you can see the user explore the picture with an aperture, revealing the face of the dog and the cat. And then in a second, you'll see a heat map, which reveals the attended locations of this image. And so that's essentially how it works. And in addition to this base functionality, MouseView gives you lots of configuration options. So firstly, you can vary the aspects of the overlay that cover the screen. You can alter the transparency. So here it is going from about 50% to 0% transparent. You can change the color to anything you want. And a big strength is the ability to blur anything underneath that overlay, even if there's a transparency of 0. And this allows participants to see the low-level features of the whole scene and use that to guide their attention. And this simulates something like the non-foveated area of our visual field. However, this is an experimental feature and can take 1 to 4 seconds to initiate, so it isn't appropriate for things like videos. We do provide a callback function, which allows you to hide the contents of the website or experiment from view whilst it generates. But things like Gorilla Zone actually already do this for you, so you don't really need to worry. The second part of the configuration is you can alter the size of the aperture. So you can change it from being very small to very big. And you can also express it as a percentage of the page width and height. And importantly, you can also apply a Gaussian blur to the edge of this, which softens the aperture. And again, this simulates some elements of foveated vision, so the edge of that foveated bit. So it's all well and me good telling you about this tool and its settings, but we wanted to find out how well this tool could replicate eye tracking results. And I'm going to give a brief outline here because others are going to talk more about this later, but I'll show you the basis of our validation paper. So our empirical question was based on a paper published by Tom and Edwin previously, which was a passive viewing eye tracking task, and it used an in-lab eye tracker. In this task, people viewed a series of pairs of images for 20 seconds, and these were either a neutral image paired with a pleasant stimuli, or a neutral image paired with a disgusting stimuli. I've used emojis here to avoid having to show you the images, but I promise you're not missing out on anything because they're quite disgusting. But you might see some of them later, so that's exciting. So when we look at the lab eye tracking results of these two types of trials, we find this pattern. We have an early involuntary look to the evocative picture, regardless of what it is, and then a later voluntary bias after this. And this bias goes in the direction of the pleasant image. So people are looking at the thing that they're interested in, and this is quite nice. And then it's flipped for the unpleasant or disgusting images. And when we used MouseView to replicate this experiment, we found this pattern. We found no involuntary look, but we suspect this may be because mouse tracking only tracks top-down attentional control, and I'm sure Tom will touch on this later. But we did replicate the later component, the voluntary bias. People dwelt on the pleasant image, but avoided the disgusting image quite strongly. And this avoidance strengthens over time, which was also the case in the lab eye tracking study. We found that this element was reliable and valid, but you can read more about that in the preprint. Okay, so like I said, we're going to get more information on what people have done with this tool later, but I'm also here to talk about how to get experiments working yourself. And thanks to the open source and general purpose nature of this tool, there are many options available to you. And these range from full-on app development to no programming at all, which is what this represents. The BeOnline.js version can be included in any app or website. And we have a Node package manager repository and a GitHub setup. And I've tried to make this as straightforward for developers to use as I can. But to do that, you do need some coding experience. Kind of in the middle between coding and no coding, you've got experiment builders like Psycho.js and PsychoPy and JS Psych. However, you still do need to do a bit of programming to understand this. But Gorilla is more towards the kind of no coding thing, so it's kind of appropriate for a small tutorial when I've only got 15 minutes. So in Gorilla, you can use the code implementation, which allows you to customize things a bit more. But you can use their task builder, and they've recently created a drag and drop zone that involves absolutely no coding at all. And that's what I'm going to show you how to use today. Okay, so we have a Gorilla zone. So this is an example of a simple trial, identical to the type of free viewing experiment I spoke about earlier. And this is the Gorilla screens in the task. You have 3 screens here. So you've got a fixation cross with a timer to kind of get a delay between each trial. And then you have a screen with a button, which is very important because it ensures when they press that button that their cursor or finger is at the center of the screen and they're not seeing one image more than the other. And then finally, the stimuli themselves side by side are shown for 20 seconds. And it's this screen that we're going to put our Gorilla Zone into. And so you select this viewing screen, and this brings up this layout, which shows the left image and the right image. And this is a little timing widget. We press the edit layout button, and this shows the layout of the screen with these gray boxes. And we want to add a new zone in, so we press that button. And this creates a new zone, which we then select with our mouse and brings up this menu where we go to the Advanced tab. And if you're on the beta program, you'll see the little mouse view box there. And this is what we press to add in. And then it's added in. And you can see it here in the center. And this will have been added with our default settings, which you can change by finding the correct section in the configuration settings. So you could scroll down. Here's the mouse tracking settings. So specifically here in the mode, record, which I'll use for this purpose, just records the mouse coordinates and does the mouse view overlay on this screen. You can use upload, which in combination with this kind of setting here, allows you to apply mouse view to your whole experiment. So you're continuously tracking over multiple screens. This then has a drop-down box with the mouse view specific settings. And in this drop-down box, you have the things I spoke about earlier. So the overlay color, the alpha, which deals with transparency, the Gaussian blur, standard deviation in pixels, and the aperture size. And these are all configurable. And now, once we've done that, we're ready to run the example experiment. So I'll play this. So when you preview the task and launch it, you will see the instructions, and then when they press next, the button appears, they click on it, and here's our blur with the aperture under. And it really is as simple as that. And this data is kind of being recorded, and you'll be able to download it in a spreadsheet that has all of the coordinates. But I don't have time to tell you how to analyze it, but hopefully we'll be releasing some code to help people do that on our website. Okay, so I think that's about it. Thanks for listening to me. You can visit MouseView.org, which has detailed documentation, links to our preprint, and examples of various different levels of implementation that I spoke about. The zone on Gorilla is in closed beta, so you can get it added to your account by contacting them using a form. I'd suggest doing this if you just want to play around with different experiment designs. It's free to make an experiment, so it's like a pretty cool way of working out what you can do. And for updates in the future, then follow me on Twitter, a bit of shameless promotion, and also follow anyone in this seminar because I think we'll all be doing things in the future. I'd like to thank Edwin and Tom, who have been great at making this project happen, but also Will at Gorilla, who led programming the drag and drop zone, and also Thomas Pronk and Rebecca Hurst at PsychoPy for implementing it there and making their examples. Okay, thanks.
Well, that was a lovely talk, Alex.
Thanks.
And really cool to see the implementation in Gorilla. Next up, we have Sonia Milani, who is a graduate student at the University of British Columbia. And she's still a PhD student, but she's done some really pioneering work on eye tracking in the context of sexual arousal and sexual dysfunctions involving low arousal. So I'm really excited about the work she's doing. And in particular, some of the work she's done has planned with MouseView. And so with that, I'm going to hand it off to Sonia.
Thank you so much, Tom. I'm just going to share my screen. Okay. My name is Sonia Milani. I'm actually not in my PhD yet. I'm actually just still finished my first year of my master's in clinical psych at UBC, but we're getting there. So I'm here today to talk to you about validating MouseView for sex research. So thank you to Tom, Edwin, and Alex for validating this so that we can use it as a tool. And so in today's talk, I will start by providing some background information to explain the role that attention plays in sexual response and review sexuality-related research that I've used eye tracking methodology to examine attention. I will then dive into describing the current study and go over the methods and results. I will conclude with a discussion of what our findings suggest, and as part of that, I will discuss our future plans for this line of research. So when we talk about sexual response, we are referring to an emotional state that is made up of physiological response, such as genital sexual arousal, as well as subjective response, such as subjective sexual arousal or excitement. Sexual function involves moving through the different stages of sexual response without difficulty. So going through sexual desire, sexual arousal, and orgasm. Not surprisingly, sexual function is an important aspect of quality of life, and studies have shown that sexual function is positively correlated with mental health, with physical health, with sexual and relationship satisfaction, as well as overall happiness. Theoretical models situate attention within sexual interactions as a key component of sexual response. This is a diagram of the emotion motivational model of sexual response. And as you can see, emotion acts early on, and it's involved in both the activation and maintenance of sexual response. This model predicts that focusing attention towards sexual cues, so for example, focusing attention on an attractive partner or sexual stimuli, this facilitates and increases sexual response. Whereas distractions that divert attention away from such sexual cues, this inhibits sexual response and decreases it. Over the last 15 years, sex researchers have used eye tracking methodology to examine visual attention patterns, given that visual attention is a central component of most sexual experiences. We see, we like, we get aroused. Specifically, eye tracking has been used to assess differences in the attentional processing of sexual and non-sexual cues, so really showing the salience of sexual cues. Studies have looked at gendered patterns of visual attention. We've examined attention as an index of sexual interest and attraction. And more recently, eye tracking has been used to investigate attentional biases underlying sexual function. So looking at differences in visual attention among clinical populations experiencing sexual dysfunction and healthy controls. And this is just to really demonstrate the utility of eye tracking in sex research. But for the purposes of this presentation, I will briefly go over findings from studies examining gender differences as it is the most relevant for the current study. For a very long time, researchers have been puzzled by a gender difference in patterns of sexual response observed in the laboratory. Studies have consistently revealed that heterosexual men show gender specificity, and what this means is that they produce a greater magnitude of physiological sexual response to preferred female sexual cues. Heterosexual women, on the other hand, show gender non-specific patterns such that they exhibit a similar magnitude of physiological sexual response to both preferred male and non-preferred female cues. Now, given that attention is theorized to be an important for sexual response, some researchers have been interested in testing whether or not visual attention is a mechanism that might explain these gender differences. So in previous eye tracking studies, and unlike Alex, I actually am using stimuli and not emojis. So bear with me, you're going to see some nude images here. When participants are shown single images of sexual male and female targets separately, results indicate that heterosexual men look more and look longer at their preferred female models. And this is a pattern that is consistent with their self-reported attraction, as well as sexual response patterns reported above. Heterosexual women distribute their visual attention more evenly and look similarly at preferred male and non-preferred female models. Women's visual attention patterns appear to be consistent, again, with the gender non-specific sexual response patterns described above. Notably, these patterns of visual attention have been found to be strongly correlated with self-reported sexual attraction ratings of the models. And so models that were male and female models that were rated as more sexually attractive were looked at more, and these effects were much stronger in men. And although eye tracking has shed important light on our understanding of visual attention patterns in sex research, this method is not without limitations, some of which Alex has already described. But specific limitations for sex research include a lack of ecological validity. Given that we are studying something that is super personal and private, people typically view sexual stimuli in private. And so doing so in a laboratory setting could potentially be uncomfortable and unnatural for people, and this could influence results. As well, the self-selection of participants who volunteer for laboratory-based sexual research may impact the generalizability, given that not everyone will volunteer to come to do an in-laboratory-based sexual research study. So to overcome these limitations of lab-based sexuality research using eye tracking, we set out to start the validation process of MouseView for sex research and conducted the current study. So the goals of the study were to examine whether MouseView would reveal the gendered patterns in the specificity of arousal that has been found using other experimental methods. And we also wanted to find out whether mouse viewing of sexual images would be associated with self-reported sexual arousal ratings. So do people look longer at what they like? In terms of hypotheses, for our first research question, we predicted that MouseView will reveal gender specificity for mouse viewing and sexual arousal, meaning that men will show gender specific patterns and women will show gender non-specific patterns. And for the associations between mouse viewing and sexual arousal, we predicted that mouse viewing will be associated with subjective sexual arousal ratings. In terms of methods, we recruited a total of 226 undergraduate students through the University of British Columbia Human Subject Pool. This included 166 women, 58 men, and 1 non-binary individual. Average age of our group was 20.5 years old, and we had a diverse sample in terms of ethnicity. Using a direct link to the online experiment, participants first completed a sexual arousal rating task. They were provided with clear images of each male and female model, and they were asked to report how sexually aroused the image makes them feel, and this was done on a scale of 0 to 10. After the sexual arousal rating task, participants completed the mouse viewing task. We used Gorilla as the experimental platform. In each trial, we presented participants with 2 images, one sexual image of either the male or female model that was paired with one neutral image depicting a random object. Participants used their mouse to move the aperture, which is the clear circle, to view the images. And here is a quick demo that I hope works. So each trial started with a central fixation cross. Once participants click that, they got to view the 2 images as they wish, and each trial was lasted 8 seconds. So to examine hypothesis 1, we looked at the gender specificity of sexual arousal, and we found that men, as indicated by the orange lines in these graphs, men showed greater specificity in their mouse viewing of the images. And so for mouse viewing, that's the graph on the left, and then for subjective sexual arousal, that's the graph on the right. So we see that men show a very pronounced preference for the female model compared to the male model, and this is true for both the mouse view as well as the subjective sexual arousal ratings. And so this is otherwise known as gender-specific response patterns. Heterosexual women, as indicated by the blue lines in the graph, show a very subtle preference for the male model. And so we can see gender non-specific patterns in women's arousal ratings as well as their mouse viewing. And then for hypothesis 2, looking at the associations between mouse viewing and self-reported sexual arousal ratings. When examining the full sample, we see a robust association between subjective sexual arousal, which is found on the x-axis, and mouse viewing, which is found on the y-axis for both the female and the male model. So the female model graph is on the left, male model is on the right. So across everyone in our sample, people look more at the stimuli they rate as more arousing. And these strong associations are observed when we look at each gender separately. So for men in our sample, we see the same positive correlations for both male and female models. And as you can see on the graph on the right, for men viewing and rating male models, most data appear on the lower end of this scale. So low sexual arousal rating and less mouse viewing. And this is expected given our mostly heterosexual identifying sample. And similarly for women, we saw positive correlations for both male and female models. And as you can see, the magnitude of the correlations is slightly lower than men's. And this pattern, again, is consistent with the gendered patterns that are observed using other methods, where the observed effects are much stronger among men than women. And so what does this all mean? Our data suggests that mouse viewing is related to sexual arousal. Our supported hypotheses replicated previous findings from studies using other methodologies, such as eye tracking and physiological measures. In terms of implications, online experimental methods applied to the study of sex, and obviously research more broadly, offer an affordable and convenient alternative to in-lab experiments. They increase our ability to recruit larger and more diverse samples. And this is particularly important in sex research because we really want to tap into populations with different sexual orientations and gender diverse populations as well. Additional benefits include levels of realism. It'll hopefully be more comfortable and realistic for participants to be viewing sexual stimuli in the comfort of their own home rather than in a laboratory setting. And our next steps for this line of research is to more comprehensively validate MouseView for sex research. And to do so, we will compare attentional processing of sexual versus non-sexual cues to replicate eye tracking findings that demonstrate the salience of sexual stimuli. And we will also replicate forced attention paradigms that have been used for preferred and non-preferred sexual cues, where participants are presented with a male and female model simultaneously. We also plan to conduct an in-laboratory eye tracking, COVID permitting, to look at mouse view and eye tracking convergence. And we are going to be doing this within samples. So the same participants that participate in mouse view will be invited to participate in an in-lab eye tracking as well. And we also wanted to examine different masks in mouse view. So we want to use the blurred overlays that I showed earlier in the demo, but we also want to examine what the data might look like if we were to flash the images clearly for 1 second to participants before the blurred overlay, just to see if one iteration of mouse view is more closely matched with eye tracking data. Thank you for listening. I'm open to some questions.
You're muted.
Yeah, so I'm back now. So that was an amazing talk, Sonia. And it's really wild to see that convergence between the subjective sexual arousal and the mouse viewing. And it's so wild too that you can do this in the privacy of your home, your dorm room, wherever, instead of coming into the laboratory, especially an advantage over some of these more invasive technologies like plethysmography. Really cool talk. So, keep the questions coming in the live chat. If you have any questions for Sonia, she might be able to address them in the last few minutes of the talk. Next up, we have Jeremy Stewart, who's a assistant professor of psychology at Queen's University and really a rising star in suicide research. And he's going to explain how some of these advantages about MouseView are really relevant to the specific population and the pressing issues that he studies. I'm really excited for his talk, and I'm going to hand it over.
Thanks, Tom. I truly wish I could see myself through your eyes. I really appreciate the generous introduction. And it seems like everybody can see this, I expect. Good. Okay, so just a couple of housekeeping items before I get started. First off, the talk is going to show some of my stimuli. And so those stimuli show people who presumably or look like they're making suicide attempts. So just as a heads up. The other thing I want to direct you to is the QR code here, which is going to provide access to a recording of the talk, my slides, as well. And I show it again on the final slide. So what I'm going to be tackling in this talk are the potential clinical applications of MouseView to better understanding and assessing suicidal thoughts and behaviors. And suicide is a serious public health concern, of course, and it claims the lives of about 800,000 people worldwide every year. And specifically, it's the second leading cause of death among mid-adolescents and young adults. Although suicide ideation, which is defined as thoughts of killing yourself, affects roughly 1 in 5 youth, only 20 to 30%, 33% of young people transition from ideation to suicide attempts. So identifying predictors of this key transition is critical for refining suicide risk assessment and for developing targeted interventions. But a lot of the putative correlates and predictors of suicide, especially in young people, they're actually strongly related to suicide ideation, but they show much weaker associations with suicidal behaviors. So in light of this issue, some modern ideation to action theories of suicide, they provide separate explanations for this initial onset of suicide ideation, and then that shift from suicidal thinking to attempts. And although these theories actually differ in their specifics, this generic model I'm showing incorporates the elements that are shared among them. So generally, the initial onset and escalation of suicidal thinking is thought to be driven by cognitive and affective variables like hopelessness and psychological pain. More germane to this presentation, though, these theories propose that suicidal actions like planning and preparation, as well as making a suicide attempt, they require the capability to die by suicide. Suicide capability refers to one's ability to overcome an innate and biological drive for self-preservation in order to engage in potentially lethal self-directed injury. Capability has some practical aspects like access to lethal means and knowledge about how to use them. And it also includes characteristics that are hypothesized to be acquired through experience. My lab has focused on an acquired aspect of capability called fearlessness about death. In these ideation to action theories, they propose that exposure to risky, dangerous, and or potentially lethal experiences, that basically habituates people to the innate fear that suicide should evoke. And then over time, people are thought to develop the fearlessness that's necessary to undertake suicidal behavior. However, evidence for that direct link between fearlessness about death, or FAD, and suicidal behavior is quite mixed. A recent review, for instance, reported that only half of the studies found this hypothesized association, and the meta-analytic effect size here was small. One reason for these inconsistent results might be the widespread practice of measuring capability with questionnaires that have poor psychometric properties. One issue is that most people can't accurately estimate or even fathom the fear they would experience if they were faced by their own imminent death. And that might undermine the validity of questionnaire measures of something like FAD. There are also several behavioral tasks that have been developed to capture capability. Just as one example, there's a suicide version of the Stroop task, and it assumes that greater interference from suicide words on performance is some kind of evidence of attention being preferentially directed to suicide content. And people have speculated that that might indicate higher FAD. But reaction time tasks like the Stroop actually don't measure attention directly, of course, and they're also prone to confounds. My colleagues and I have also recently found that interference on the suicide Stroop has low internal consistency and poor test-retest reliability. So we were very fortunate to team up with Tom Armstrong and his lab to improve the measurement of FAD by using eye tracking. Dwell duration recorded by an eye tracker, as many of you might know, yields more differentiated responses to unpleasant stimuli than other behavioral or even psychophysiological measures. And so here it might capture suicide-specific responding with greater precision. Dwell is also a more direct measure of overt attention, and it has strong psychometric properties. So we used a similar simple passive viewing task that you saw in the prior talks, in which participants see pairs of emotional and neutral images, like the pair I have shown here. The emotional images included suicide-related, threatening, pleasant, as well as disgusting stimuli. And there were 5 images representing each category. Each emotional image gets shown 4 times for 12 seconds in each trial. And we record dwell duration, defined as how long gaze is fixated on each image with an eye tracker. Our validation study recruited 140 students, and we oversampled for prior suicidal behaviors. So the sample did include 28 people with a prior suicide attempt, and 10 additional students reported making an interrupted attempt. Along with several questionnaires that captured demographics and psychiatric symptoms, participants completed the suicide ideation scale, which measures the intensity of thoughts of suicide in the past week. That measure actually has 2 separate subscales. On the one hand, there's suicidal desire that reflects hopelessness and kind of a non-specific desire that life would end. And the other subscale is called resolve plans and preparation, and that includes thoughts and actions that indicate greater readiness to make a suicide attempt. So turning to the results, this figure depicts gaze behavior on suicide image trials. The lines basically represent how much participants were fixating on the suicide images relative to neutral. So higher positive values indicate longer dwell time on the suicide image. The trial time is divided into 12 one-second epochs on the x-axis, and the separate lines show the patterns for each presentation of the image from first to fourth. So what you can see in the figure basically is that there's a significant linear decrease in dwell on suicide images relative to neutral images across the epochs. What we've also found and reported in this paper is that gaze behavior towards suicide image seems to be distinct from the patterns of viewing we see for other emotional images. And we also found that participants who rate the suicide images as subjectively more frightening and more disgusting, they tend to dwell less on suicide images compared to the neutral images over the course of trials. So that supports the possibility that our task captures cognitive affective processes that are relevant to suicide capability or fearlessness about death, or both. We next examined whether gaze behavior towards suicide images was associated with individual differences in recent suicide ideation severity. So in this figure, you can see scores on resolve plans and preparations on the y-axis. And on the x-axis, there's a measure of overall gaze behavior that we use a lot called the proportion of dwell time on suicide images. And this score ranges from 0 to 1. With equal viewing of the 2 images, the 2 categories of images, yielding a score of 0.5. And that's shown in the green dotted line here. Scores above 0.5 indicate longer viewing on the suicide images. So what the figure shows then is a small but statistically significant association between dwell towards suicide images and resolve plans and preparations. And given this substantial skew you can see depicted here in the outcome variable, we also fit a regression model with several demographic and clinical covariates, and we used a percentile bootstrapping approach. The effect remains statistically significant. Also, dwell duration on the other emotional images wasn't significantly related to resolve plans and preparations. We conducted the same analyses on suicidal desire, and that's depicted here on the y-axis again. The proportion of dwell on suicide images wasn't bivariately associated with suicidal desire, and the effect was also non-significant in the multivariate bootstrapped regression models. In fact, dwell behavior in general, regardless of the category of emotional image, wasn't associated with suicidal desire in our sample. Turning now to participants' history of suicidal behavior, this figure has the proportion of dwell on suicide images, this time on the y-axis, and the number of prior suicide attempts on the x-axis. The boxes represent the distribution of dwell within each bin of lifetime attempts. So median dwell towards suicide images, which is represented by the dark lines dissecting each box, you can see that it increases with the number of prior attempts. And in fact, when you analyze that in a negative binomial regression model, as we did, greater dwell time on suicide images is associated with a greater rate of past lifetime attempts. Finally, we investigated suicidal intent among the 28 suicide attempters in our sample. In line with what ideation to action frameworks would predict, greater dwell towards suicide images was associated with higher self-reported suicidal intent among the attempters. I'm suspending major inferences, of course, until this is replicated in a much larger sample. But again, we find that the effect for suicide images was not significant. The effect on intent was not significant for any other type of emotional image we used. Okay, so to summarize, the participants in this study looked away from suicide images over time when they had the option to look elsewhere, at least on average. However, those who rated suicide images as less aversive dwelled on them longer. So this pattern of results suggests that our dwell time measure might be capturing some important individual differences in something like fearlessness about death. Like other aversive or unpleasant stimuli, suicide images could initially capture attention, but ultimately evoke perceptual avoidance over time. Those with greater FAD, for instance, might engage in less ocular motor avoidance than people with lower FAD. Greater dwell on suicide, but not any other type of emotional image we used, was associated with suicide planning and preparation, with prior lifetime attempts, and with suicide intent among those with a history of suicidal behaviors. Returning to that ideation to action framework, dwell on suicide images was specifically associated with behaviors that portend high lethality suicide attempts or dying by suicide. Ultimately, this type of measure could have immense clinical utility for guiding treatment and follow-up decisions for suicide ideators in psychiatric settings. Of course, COVID-19 really forced our hands and necessitated an online solution for keeping what I think is a promising line of research going. Fortunately, as we've seen today from both Alex and Sonia, there's already exciting evidence that attention tracking in MouseView is strongly associated with dwell time measures recorded with eye tracking using similar free viewing tasks as we did. So we're currently running a study that's hosted in Gorilla, in which we present pairs of images like the ones to the bottom left of the slide here, and we record gaze with MouseView. So far, we've recruited 170 participants. We have 69 with a history of suicidal thoughts and behaviors, and we expect to meet our target sample of 250 in the fall. So far, I wish I had data to report. I don't, but the task is intuitive and it's acceptable to most participants. The percentage of unusable trials is similar to our in-person study. And once the study's complete, we're excited to test whether the MouseView version of the task shows the same pattern of associations with recent suicidal thoughts and lifetime suicidal behaviors as we saw in our in-person study. If that indeed happens, and we find our MouseView, the MouseView version of the task is sensitive to key indicators of suicide capability, there are important implications for just how we study suicide. Suicidal behaviors, of course, have low base rates in the general populations, and so studies require very large sample sizes to conduct the complex and detailed research that we need to inform prevention. Because of this, most research investigating behavioral and neural correlates of suicidal behaviors in particular uses small clinical samples in which the rates of suicidal behaviors are much higher, or they enrich smaller samples by oversampling for suicidal behaviors like we did. The limited statistical power in many studies likely contributes to a modest expected replication rate for studies on suicidal thoughts and behaviors. I have a former student named Brendan Lamb, and he's working on a project that directly assesses the replicability of work in our field. And so far, he estimates that about 2/3 of existing studies would replicate, and he also finds evidence for an inflated false discovery rate in these studies. I think MouseView provides an especially promising solution for this issue. So if we find evidence that this task is a valid measure of suicide cognition, the online delivery format is scalable so that it could be administered to nationally representative samples of thousands. That would yield critical information about the generalizability of our measure and its utility for understanding and predicting suicide risk. We think, at least preliminarily, that this is quite feasible because of the success that other research groups have had with administering online behavioral tasks to large samples. For example, Project Implicit, which is led by Bethany Teachman, Matt Nock, and several others, actually administered a death-suicide version of the implicit association test to volunteers through their website. And I think that we could validate our MouseView task in a very similar way. So I'll leave it there. I'll end by acknowledging funding sources for this work, my students, and of course, my collaborators, including Tom. Thank you.
Thanks, Jeremy, for your fascinating talk. I'm not only excited about your dwell time findings, but the case you make at the end for the role of MouseView in robust, reproducible science of suicidality is really strong, and I'm excited to see you scale up this dwell time research using MouseView.
Yeah.
I'm going to conclude these talks with a little discussion. It's my first time in the discussant role. I'm pretty excited. Let me get my screen share going, and I'll tell you a little bit about how I think we can integrate these talks. All right, so I'm going to be talking about what it is exactly that MouseView.js measures and why it matters. And I'm going to be focusing on the context of these passive viewing tasks. And that's the only nasty image I'll be showing today. So yeah, I'm asking the question, what is MouseView measuring in these tasks where people are asked to view an emotionally charged or motivationally relevant stimulus, and they're presented with the stimulus alongside a neutral image for about 6 to 10 seconds. So one thing we see in these tasks that Alex highlighted is that there appear to be two components of gaze. There is this early automatic bias. Move that window out of the way. And then that's followed by this more voluntary bias that plays out over the course of the trial. Now, most of the research so far has been on this early bias. The construct of attentional bias, it's often focused on this early, relatively involuntary allocation of attention to emotionally charged and motivationally relevant stimulus, regardless of their valence. This later bias hasn't been studied very much. It's been greatly under-theorized because you can only really study it with eye trackers. The thing that's great about this later bias is that it's quite reliable. It has good internal consistency and retest reliability, whereas the early, more involuntary bias is hard to measure in a consistent, reliable manner. So, as Alex pointed out, MouseView seems to mainly measure this more voluntary component of gaze. And that makes sense because mouse movements are more effortful and they're slower than eye movements. So mouse viewing is a proxy for voluntary overt attention. But what determines where we voluntarily look? What is it that we're ultimately measuring in these passive viewing tasks that you've heard about today? Well, one appealing solution, because of its parsimony, might be to assume that looking is determined by which motivational system is activated and to what degree. So appetitive motivational circuitry and sort of a more general description of the motivational systems in humans and other mammals. Appetitive motivational circuitry motivates approach behavior towards beneficial stimuli, whereas defensive motivational circuitry motivates avoidance behavior away from noxious or harmful stimuli. And it may be the case that you look towards things that activate this appetitive circuitry, you look away from things that activate this defensive circuitry, and the degree of looking towards away just depends on the potency of the stimuli. However, this does not entirely account for looking behavior in these tasks. In fact, it really doesn't map on that well. And as Jeremy and Edwin Dahlmeyer and I found, it's really only disgusting stimuli that cause people to really reliably look away from an emotional stimulus compared to an accompanying neutral stimulus. And people actually don't actively avoid rather unpleasant, rather potent stimuli such as images depicting suicide or images of threat and violence. So this raises the question, why would you look towards something unpleasant? Why do people look at noxious, potentially nasty things? And one answer is that they do so to learn more about these stimuli. The emotion of interest or curiosity has been hypothesized to have an appraisal structure of novelty and the potential for understanding. You see some roadkill from a safe distance. That's something new. You've never seen the insides of a raccoon, for example. And within the mess, there is some order. You can make out some organs and things like that. So this is one of the reasons, kind of a flippant example, but why people would dwell on and look at something unpleasant. And we actually even see this for disgusting stimuli, even though overall these sort of uniquely prompt oculomotor avoidance. So as you can see in this first presentation with eye tracking, dwelling on the disgusting images continues into voluntary gaze. And what's really nice about MouseView is we capture this same effect where people first sort of check out and explore voluntarily unpleasant, even disgusting images. And I think, turning to a more serious, pressing matter, this could help us understand why folks, particularly people high in suicidality, would look more at images of suicide. And so there may be this interest or curiosity, due to the lack of fearfulness of death that Jeremy described. It could also be related to negative reinforcement. So some theories of suicide ideation posit that the thoughts sort of function to relieve distress and offer a sense of perceived control. So negative reinforcement may be a factor as well. Why is it that we look away from something unpleasant? Well, as I've noted before, this seems to be pretty unique to disgusting stimuli. It's also been observed for undesired nudes that people rate as disgusting. And I think this might provide some insight into the nature of disgust, that disgust involves stimuli that are inherently revolting, unpleasant to contemplate and perceive. But one enduring question is whether or not oculomotor avoidance of disgusting stimuli is part of disgust itself, the emotional response, or a later sort of instrumental behavior that's an emotion regulation strategy. And it might be somewhere in between. It might be just a deeply ingrained habit that's entrenched by negative reinforcement. So another open question. Turning to Sonia's talk, we can also ask why it is that people look towards something pleasant in these tasks. And a simple answer is that it's reinforcing. Here is that model that we saw earlier that's been revised by Samantha Dawson to better sort of parse attention. But controlled attention leads to self-reported arousal and also genital arousal, so things that are pleasant. So sustaining dwell on sexual images can be reinforcing. However, I think there's an open question here of whether dwell on sexual stimuli reflects liking or wanting. So I'm turning to these constructs from Kent Berridge in the incentive saliency model of dopamine. And we know that the sexual motivation piece, this wanting, isn't always pleasant. So cravings, desires, not just for sex, but for drugs, for food, they are actually sort of orthogonal to pleasure, and they often aren't pleasant. And so one open question is whether that wanting, when it's unpleasant, still leads to dwell on the sexual stimuli. And finally, I want to raise another possibility about looking away from things that are pleasant. And this is actually something that Professor Richard Lopez at Bard College is interested in studying with MouseView. And so we're talking about using MouseView to look at potential self-regulation strategies to resist temptations and manage and reduce cravings related to food, alcohol, and other stimuli. So in summary, looking matters in clinical and affective science. It's been understudied, it's been under-theorized, but it reveals clinically important information. And as Jeremy has shown in relation to suicidality, as Sonia has shown in relation to sexual dysfunction, and in other data that we haven't presented today, we found that mouse viewing and eye tracking really reveals specific phobias. Also, looking is not just an indicator, it's also an important causal process. And so Sonia, in some of her more recent eye tracking work, Flory Brado, has shown that maintaining gaze on sexual cues is important for sustaining sexual arousal. So this could be a potential treatment target. And this is actually a line of work that Professor Samantha Dawson at the University of British Columbia is planning to work on, work that will involve MouseView. So in summary, I'm really excited about MouseView because of its potential to accelerate research into this important yet understudied phenomenon of looking. And one of the things that makes me so excited is that I've collected more data with my collaborators in 6 months using this method than I did in 6 years using eye tracking. So I'll leave you with that thought. And I just want to thank the good folks at Gorilla, Joe and Josh, for supporting us as we developed MouseView. This was the platform we used for all of these validation studies. And I want to thank my friends and collaborators who helped build this tool and get it started.
Tom, you're more than welcome. We love helping researchers like you, and thank you to Jeremy, Alex, and Sonia as well for sharing your research. That was a heady move from sexual arousal to suicidal ideation. That was quite a contrast, but both of you, I think Tom's managed to extract the overall meaning out of that so well. And I loved how Jeremy ended his talk talking about the journey from reproducible science to generalizable science to impactful science, and how the work that we're doing as behavioral scientists is really important. Jeremy's work could ultimately save lives. This isn't just for fun. This isn't just for discovery. We have a chance with the subjects that we choose to study to save lives, to change lives, and that's really important. And if you want to hear more about that, then come to the session about reproducibility tomorrow afternoon.